Welcome to today's webinar. Welcome everybody. We'll give people a couple minutes to come in and settle before we start. All right, 2 minutes in. So with respect to everybody's time, we'll get the webinar started. My name is Zhiying Li. I'm the Director of Life Science and Technology at GemPharmatech US. It's my pleasure to bring Santi again to the webinar for today's delivery. So let me share a brief intro of Santi. Santi is a research scientist and business development professional over 10 years of experience in autoimmune disease, immunology, and oncology. She received her training in human B-cell development at the Government Institute of Medical Research, Australia, in immunology, followed by a postdoctoral training at the Children's Cancer Institute, also in Australia, focusing on pediatric leukemia and therapies. Her projects included efficacy evaluation and mechanistic investigation of a variety of testing agents, such as navitoclax, venetoclax, and selumetinib. Her research projects resulted in multiple publications in journals such as Blood and Journal of Immunology, as well as national and international awards. Santi joined GemPharmatech as Associate Director in Immunology and Oncology. Today, she'll be sharing some new insights and data on KPC mouse model and cell line that GemPharmatech has developed to support the pancreatic cancer research and therapy development. So without further ado, Santi, the stage is yours. Thank you.
Thank you, Zhiying. Let me share my screen. Okay, there you go. Great, thank you everybody for attending today's talk. It's my great pleasure to discuss about one of the model that's currently available at GemPharmatech. So just a little bit of disclaimer about our company here. So an outline of today's talk will be, firstly, I'm going to give a brief overview of our company and then the offerings that's available through GemPharmatech. And then specifically for today's talk, it's about pancreatic cancer and the Kras mutation. And then I will highlight the two models. One is the KPC mouse model, which has the Kras G12D and P53 mutation that's available either as a mouse or we've also developed a cell line out of it. And I will talk the second model with the Kras G12C, which is the recent addition to our catalog. And I'll show you some preliminary data on this. So GemPharmatech is a one-stop solution for biomedical research. We have the largest collection of genetically engineered mouse model. It's all from one organization, and we make it available to the R&D community to support their effort to understand more about various human diseases. So as I mentioned before, we are the world's leading mouse model repository. We have over 22,000 different models that ranges from conditional knockout, humanized model, immunodeficient models, germ-free resources, all of it available for a variety of therapeutic areas. And in addition to all of the mouse model, we also have a full suite of preclinical testing and pharmacology service. So if you're interested for us to do all the testing for your agent, it's available in our company. So we are a one-stop solution for preclinical research where you can either purchase the mice or we make the mice, we'll do the breeding, and then we'll also do the testing. In terms of therapeutic areas, it's summarized here. We have immuno-oncology, spontaneous tumor, which I will dwell deeper on for today's talk. But we also have autoimmune, COVID-19 research, rare disease model, neurodisease model, as well as metabolic disease model. So I invite you to scan this QR code here on the bottom right-hand side, where there are past webinars that have highlighted certain models as shown here. So for today's talk about pancreatic cancer and Kras mutation. So pancreatic cancer, obviously, as the name suggests, begin in the pancreas, as shown on the left-hand side here. And on the right-hand side is the 5-year relative survival rates, as shown by different stages and of diagnosis and race. It is quite dismal if you're diagnosed with pancreatic cancer, approximately less than 50% survival rate, as shown here, if it is a localized cancer. If it is regional, distant, and also and all the variety of stages, it's less than 20%. So we need a better treatment for pancreatic cancer to improve the survival rate. Various studies have done to understand the mutation that's involved in pancreatic cancer, as shown here. I don't want to go deeper into this, but just want to highlight, for the purpose of today's talk, I want to highlight the Kras and TP53, which is the major driver of pancreatic cancer. So within pancreatic cancer, obviously Kras is an important mutation, but just to take a little bit step behind, RAS is the protein, and there's multiple isoforms, of which Kras is one of the isoforms here. The other isoform is the NRAS and HRAS, which is also implicated in a variety of cancer, as shown here on the left-hand side. Here we can see that in melanoma, NRAS is about 29%. Meanwhile, for pancreatic, for today's talk, it's the Kras, which is about 86%. Within each of the isoform, there is point mutation as well, which is summarized here on the right-hand side. For the pancreatic, is 91% is in G12, either G12V, G12R, G12D, and some of the others. For the colorectal, we can see here 68% is in G12 again, with V, C, and D playing an important role. And down the bottom here, we can see the lung as well as melanoma, which is the NRAS isoform. Various companies have developed therapies that targets within the RAS signaling pathway. As shown here, we have the EGFR inhibitors, we have the AKT, the mTOR, the MEK, MERK, and RAF. So we want to create a model where you can test the efficacy evaluation, specifically that involves the Kras G12D. And that's the goal of GemPharmatech and for today's talk as well. As I mentioned before, not only Kras, TP53 is also an important mutation, and both P53 and Kras is required to generate the pancreatic cancer and metastasis in mouse model. So for today's model spotlight, we created the B6KPC mouse model. And this slide here showing the strategy, we created a mouse called the B6 Kras G12D, which then is crossed with the B6P53 point mutation of R172H. And then from here, we then cross the resulting mouse, which is the B6KP, with a PDX-Q, which is specific for pancreas. So the term we call it is the B6KPC is the result of all of this crossing. So to highlight, I will speak about this first mouse, which is the B6 Kras G12D to begin with. This is the strain information if you're interested in purchasing the Kras G12D. It is on a, if you see the LSL, it is on a stop locks system, so it needs to be crossed with a Cree to activate it. And the strain ID, which is unique to GemPharmatech, is for this strain is here, T004551. When it was crossed with a lung-specific Cree, so LYZ2 Cree, so you can see here, we can see the development of the tumor within the lung. When we cross it with the liver, the ALB Cree, you can also see here, as shown by the arrows and the stars and the triangles, we can see the development of tumors both in male and female at about 36 weeks. So there's really a lot of application on how this mice can be used if you want to cross it with a variety of different Cree for organ or tissue specific. So moving on to the model of the P53 mouse. Again, this is the strain ID if you're interested to purchase the P53 point mutation R172H. It's T007671. So when we crossed these two, it resulted in another mouse that's denoted by B6KP that has both the TP53 and the Kras. And again, the strain ID is the T01583. For the purpose of today's talk, this is further crossed with the PDX-Cree to make it pancreatic specific. As shown here in the H&E staining, the B6KPC developed the spontaneous tumor in the pancreas at about 10 to 12 weeks. It can range to about 16 weeks. As shown here in the arrows, in the black and the red, you can see that there's the tumor cells coming up. It does metastasize as well, in particular to the lung here. One of the downside of this is the slow onset of pancreatic cancer. It ranges between 10 to 16 weeks. So it's a little bit unpredictable, which makes it hard to monitor. And because of that, we make a cell line out of this to make it more predictable for preclinical efficacy evaluation. So for the cell line, we call this the mPAKPC cell line. So this slide here shows you the step-by-step process. This is the B6 KPC, spontaneous pancreatic cancer. We took the cancer out and then we subcutaneously inoculated into a naive B6 just to make sure it grows. And then we take it back out again and then make a single cell preparation, infected with luciferase to make it for the purpose of further autotopic inoculation. So here in step 4, we inoculated in the pancreas, and as you can see here, we can see a nice growth using IVIS imaging. Then this particular cell line then can be used for efficacy study in an autotopic fashion. Showing you some of the validation of this cell line is looking at the tumor volume. So for this particular one, it was subcutaneously injected. You can see a nice tumor growth here. We also confirm the P53 and the Kras mutation via sequencing, as shown here on the right-hand side. We then do in vivo growth kinetics. As you can see here, autotopic inoculation. You can see from day 7 till day 28, you can see a nice increase growth of the tumor. And it's summarized here on the right-hand side, looking at the radiance. We can start detecting it from 7 days, and it can be continuously used over time. We also look at the tumor infiltrating lymphocyte. It's within the immuno-oncology space. This is a crucial factor. So we look at the variety of like T cells across 14, 21, and 28 days. And you can see the changes in the TIL infiltration on the T cell, looking also the CD4, CD8 here in the middle, as well as the myeloid down the bottom here. As I mentioned earlier, this is also a good model for metastasis purposes. You can see here across day 14, 21, and 28. This is within the tumor, but you can see within the lung, you can see the tumor as well within the lung. So this is a nice model for pancreatic cancer that is predictable. So you can do autotopic inoculation. You can easily monitor the cancer progress using an imager. And we know that it metastasizes to the lungs, so that's nicely to recapitulate what happens in humans as well. So to show you the application of this model, a case study. So this was tested with a gemcitabine, anti-CD40 here, and also the combination of gemcitabine and anti-CD40. You can see the overall tumor weight here, the vehicle as shown in the black. We can see there's a nice tumor growth inhibition with gemcitabine therapy. But not with anti-CD40 by itself. You can see a nice synergistic effect when both gemcitabine and anti-CD40 is combined. And at the end of the study, we excise the tumor, and you can see here the nice photograph of the tumor. So a second case study, we tested this particular model, this time with a humanized PD-1 and CTLA-4. And as you can see here, again, on the left-hand side is the tumor weight across a variety of treatment. And on the right is the radiance. We tested it with the IMAP362 and also the Yervoy and also in Keytruda and in combination of this. I'm going to highlight here on the right-hand side, looking at the radiance. We can see that there's the therapy definitely showed some kind of a tumor growth inhibition within the first week post-treatment. And it continues until around about 15 days. And after this point, it seems to grow and doesn't show significant tumor growth inhibition by the end of the study. So again, this is a nice evaluation, and it actually translates to the clinical, where we see, for example, with Keytruda, there's not really a clinical response. So part of the characterization, if you would like to know a certain expression, for here we have done the Claudin 18.2, CD73, PD-L1, and CD47. And you can see here that the mPAKPC cell line expresses the Claudin 18.2, CD73, PD-L1, and CD47 quite nicely here. This is also an example where we can analyze the binding activity for certain antibody that targets these proteins as well. So just a quick information of the newest model, which is the Kras G12C model. As I mentioned before, there's also a Kras G12D inhibitors that's available in the various clinical development. So because of that, we make the Kras G12C so that you can do a proper evaluation on the preclinical stage. And again, this is the strain ID that is unique at GemPharmatech. So if you would like to purchase it, please quote the T051658. And this is the preliminary data. When it was crossed with a lung-specific tree, it developed tumor at 3 weeks. Here you can see that there is the congestion of the blood vessel here, and also the inflammation as shown by the red and also the yellow arrows. So this model can also be crossed with other creams if you're interested in other organs, such as whether it be pancreas. We're also doing some more internal validation with this model. So I invite you for a conversation with us if you would like to know what's the status of this model. Not only on the Kras, we also have other spontaneous tumor model as summarized here in this table. As you can see, we have the APC MINT, which is the intestine tumor models. We also have the breast cancer model. We have the global p53 knockout, if that's of interest. Variety of the Kras, G12C and G12D as discussed today. We also have the humanized MYC that can be crossed with CRE. We have the SMAD4 and the P10. And in red here are the variety of spontaneous tumor model that's currently under development. At the heart of GemPharmatech, we make more and more mouse model, and we're interested to speak to you if you want to make a custom spontaneous tumor model. So just want to quickly highlight again the different therapeutic areas that's available at GemPharmatech. Today I've discussed about the spontaneous tumors, specifically about the Kras G12D and G12C. But we, as mentioned earlier, we also have a huge portfolio of IO models, autoimmune models, COVID-19 models, rare disease, neuro, as well as metabolic disease model. Just a quick summary. So today I've highlighted two models, the Kras G12D, that's been crossed with P53R172H. It's available as a mouse as well as in cell line if you're interested to use this for autotopic or subcutaneous inoculation for preclinical evaluation. We also have the Kras G12C model. If this is an interest, please do contact us, and our details is here. And I invite you to visit our website at www.GemPharmatech.us to have a look at all of our other catalog of mouse model. Thank you very much.
All right, that's pretty short and sweet. So I invited everybody to put your question if you have in the Q&A box. So I'm gonna go there and take a look. So I see two very short technical questions for Santi. So the first one is talking about the luciferase tagged cell line versus the non-tagged cell line. Have we seen responses coming from the luciferase tag?
Yeah, we did a comparison, and the growth with or without luciferase is the same. So that suggested to us that there is no additional response that's coming due to the luciferase tag. However, if you want to do I guess a proper immunogenicity evaluation, then we need to have a look at the cytokine and then look deeper into the TIL and the potential different activation that occurring inside. We haven't done that yet.
Great, thanks. I have a question here. How did you measure the tumor weight? Are there multiple tumor nodules in the lung?
Yeah, so that's usually taken at the very end. For the pancreatic model, it's actually quite easy to take it out. So we just, at the end of the study, then we would cull the mice and then separate the tumor from the pancreas and then measure it that way.
Mm-hmm. Another a bit technical on the detail. So you have shared data on the metastasis into the lung. Have you looked into other tissues?
Yeah, we have only looked at the lung at this point. We haven't do a comprehensive look at the other organs yet. We did not suspect, I mean, we also base it on the IVIS imaging, and that's where the radiance is. So that's why we only focus, but we haven't done a full body evaluation.
All right. One more question about the KRAS G12C. You shared some data. Do you have more data that you can provide for this particular model?
Yeah, this one is a recent addition to our catalog. We are, as I mentioned earlier, we are doing more analysis and crossing it with more pre, just to do more validation. Hopefully in the next 1 or 2 months, we should have more information.
Great. I think as far as I can tell, there's only one last one. So you showed some efficacy data on using Yervoid and Keytruda in the mice. How do you, in your opinion, how is that reporting the clinical observation so far that had been observed among the patients?
Yeah, so that's a great question. The clinical trial shows no effect in the unselected patient with advanced, pretreated, and progressive pancreatic cancer. In our model, the Keytruda showed some kind of anti-tumor efficacy. As if you still remember, there was a dip in the first 14 days, but then it did increase over a span of another 2 weeks. And at the end of the study, there was no difference to the control. So this is, in our opinion, is translatable to what is seen in the patient. So that's why this is in our opinion, is a good model for preclinical evaluation.
Got you. One last question just came in. For the cell line, what strain do you need to grow them in?
Yeah, this cell line is created from a black 6 background, so you would need a black 6 mice for it.
All right. One last check in the chat box. I think we have answered every question that we have received. So thank you so much, Santi, and thank you so much for everyone who joined us today. We'll see you next time in November.
Thank you, everyone.